Hello everyone, my name is Emma DURFEE and I'm here with Ava Deanda, and today we'll be presenting our research on the use of human blood as an additive in chocolate agar plates, particularly focusing on its effectiveness for antibiotic susceptibility testing in clinical settings. The title of our study is Human-Packed Red Blood Cells as a More Accessible Blood Agar Additive for Use in Resource-Limited Areas.
Human blood is not commonly used in the production of blood media, which traditionally relies on animal sources such as sheep or horse blood. However, in some countries, access to animal blood can be limited, making human blood a potential alternative. The lack of standardization in preparing human blood media has posed challenges for laboratories, particularly in cultivating and identifying fastidious microorganisms.
Our research builds on previous phases that optimize the use of human blood for producing chocolate agar plates, or CHOC. Our current focus is on testing antibiotic susceptibility to ensure that human blood-derived chocolate plates provide reliable and reproducible results in a clinical setting. We hypothesize that human blood can be effectively used in chocolate agar plates and that the results for antibiotic susceptibility will be comparable to those obtained from traditional sheep's blood chocolate plates. The pictures included on the poster display the process of making the human blood agar, obtaining results by measuring zones of inhibition, and examples of growth on the 3 different media types with antibiotic discs. A closer look at the plates that Ms. Deanda and I made are provided below the discussion section of the poster.
To conduct our experiments, we used expired blood products from local hospitals, commercially manufactured sheep's blood chocolate, Haemophilus test medium, and antibiotic discs. Our process involved using an autoclave, sterile docker, and a nanopure water purification system to prepare human chocolate media. The methodology is a continuation of previous phases. Phase 1 validated the minimum concentration of expired human blood required for blood agar plates. Phase 2 developed a method for producing chocolate agar to grow fastidious organisms. Phase 3 adapted this method for resource-limited settings using accessible materials and confirmed the autoclave as a suitable device for lysing red blood cells. Many fastidious microorganisms, such as certain species of Haemophilus and Neisseria, require specific nutrients found in red blood cells, which are more readily available in lysed blood. Lysed blood releases these nutrients into the agar, making them accessible to the microorganisms. For diagnostic purposes, lysed blood media can also aid in distinguishing between different types of bacteria based on their hemolytic patterns. This can be especially useful in identifying and classifying bacteria in clinical samples. In the current phase, we compared antibiotic susceptibility results on homemade human chocolate plates, traditional chocolate plates, and HTM plates. We used a 0.5 McFarland standard inoculum of Streptococcus pneumoniae, Haemophilus influenzae, and Klebsiella pneumoniae. Each plate was tested with antibiotic discs, ceftazidime, piperacillin, amikacin, norfloxacin, and cefotaxime. The zones of inhibition were measured and compared according to CLSI M100 standards.
Table 1 presents the zones of inhibition for the various media, manufactured chocolate, human chocolate, and Haemophilus test medium. Notably, Haemophilus test medium is specifically designed for isolating and identifying Haemophilus species, so its comparison is primarily relevant to this genus. Our results indicate that human blood media yields accurate and reproducible results for antibiotic susceptibility, similar to those obtained with traditional sheep's blood chocolate.
Table 1 shows that using human blood media as a substitute for animal blood provides reliable results for susceptibility testing of different organisms. This suggests that human blood could be a viable alternative in settings where animal blood is not available. However, to ensure accuracy and reliability, additional replications are needed. Our goal is to achieve 20 consecutive replications in line with CLSI guidelines to meet U.S. clinical laboratory standards. In summary, our study shows that using human blood in chocolate agar plates is a viable option for clinical use, especially in areas with limited resources. This approach can make it easier for labs in these regions to perform important tests. However, we still need to do more testing to confirm these results and make sure This method is fully ready for use in everyday clinical settings. Thank you for listening, and we'd be happy to answer any questions you have.